Welcome back, guys. This is Falgun and we are here again. Today we are going to discuss about the embryo development, right? Embryo development. As we know, this particular stage, embryo development, okay, this particular stage of development occurs just after fertilization, as zygote will develop into embryo. So that is what we are going to discuss. So the developmental stages, or the development stages of embryo, okay, that developmental stages of embryo is called as an embryogeny. Understood? So first and foremost thing that we need to understand, that is embryo is developed from zygote by multiple divisions or by multiple mitosis, right? So what we actually know, zygote is present at micropylar end of the embryo sac, right? So the development of embryo also takes place at the micropylar end of the embryo sac where the zygote is situated. So we are clear with this first point, right? So coming to the second point, what it is given here is the early stages of embryo development are similar in both monocotyledons and dicotyledons. If you don't know, then I'm here to inform you that embryo are of two types, monocot embryo and dicot embryo, that we'll be discussing in coming up slides. Okay, so stay tuned. So guys, here what you're looking into is the fertilized egg that gets divided and it forms two cell stages. Then again it gets divided continuously to form the various amounts or the various stages. At last what we get is the mature embryo. Understood? So this is how the development of embryo takes place. Okay, so mature embryo may be of two types, that would be like that is dicot embryo and monocot embryo, that we'll be discussing in coming up slides, as I mentioned. Okay, so but early stages of the embryo development, that is embryogeny, will be same or will be similar in monocot embryo and dicot embryo. Okay, later stages will be varied in the monocot and dicot embryo, that will be concentrating while we'll be discussing about the structure of monocot and dicot itself. Okay, so without wasting any time, let's move on to the next part, that is dicot embryo. Okay, so let us discuss about it. Let us move on to the first point, what it is offering us to learn. So the first point says, so dicot embryo is enclosed in between two cotyledons. Okay, as you can see, these two are the cotyledons, cotyledon number 1 and cotyledon number 2. Let me just change the color of the pen, okay, so that it will be more easily visible. 1 second. Yes, this is cotyledon number 1 and this is cotyledon number 2. Okay, so it is present in between it, so no problem, right? So dicot, the name resembles, it has two cotyledons, no doubt in it, right? Apart from that, what we got in this dicot embryo is it has a plumule, and this plumule is what we call it as an shoot region. What it don't have is the protective sheath. So dicot embryo don't have any protective sheath that protects plumule and radicle. Okay, so in simple terms, if I speak, plumule and radicle lacks protective sheath, and dicot embryo also don't have scutellum. So many of you might be wondering, what is the role of scutellum? Scutellum is nothing but scutellum basically helps to absorb the nutrition from the endosperm. Okay, as we know, dicot embryo doesn't have endosperm. Okay, that is the reason why there is no scutellum, or scutellum is reduced in this structure, because there is no endosperm is present in this structure. So coming to the next slide, that is about the monocot embryo. So what it offers us to learn, let us look into it. So monocot embryo is enclosed by the single cotyledon. Okay, mono is nothing but mono resembles single, or mono resembles one. Here monocot resembles one cotyledon. In that case, we have understood that dicot resembles two cotyledons. Here monocot resembles one cotyledon. One cotyledon embryo. Nothing but it's very simple. As simple as it is, as you can see. Okay. As a result, now let us look into the structure of monocot embryo. So monocot embryo has a plumule in the lateral position. As you can see, this portion of this region is called plumule, as it has written shoot apex. That is what we call it as a plumule, okay, or future shoot. This region of the plumule, this region where plumule is situated, that is called as a lateral position, okay. So monocot embryo has the protective sheaths, okay, as you can see in this diagram. It has protective sheaths, and that protective sheaths are called as an coleoptile, that covering the plumule, and coleorhiza, which covers the radicle. From this, what we have understood, once again I am repeating. See, coleoptile covers the plumule, that is shoot apex, or coleoptile covers the shoot apex, and coleorhiza covers the root apex or radicle, okay. So that is what we have learned, that unlike dicot embryo, monocot embryo has the protective sheaths for the protection. So monocot embryo has a scutellum. So as I had mentioned the role of scutellum, again I am mentioning it, no problem in it. So scutellum basically absorbs the nutrition from the endosperm which is present in it, or which is present in the embryo sac. The main role of scutellum is that, okay. So here in this monocot embryo, we can able to see the reserved endosperm, okay, which is helpful to nourish the embryo. So this scutellum basically absorbs the endosperm and nourishes the embryo. So that is about the embryo. Thank you for joining in. This is Falgun. If you really like this particular video or most of my videos, then consider subscribing, so that it gives me confidence to make more videos. That is it for today. Falgun signing out.